Assalamu alaikum warahmatullahi wabarakatuh. So for 15 minutes, I will try my best to deliver the message. I want everyone to leave from this talk knowing what is combinatorial chemistry. Combinatorial chemistry. So drug discovery. So we go here. So when we speak drug discovery, so with the high throughput screening and the mapping of the human genome, which means we have 30,000 genes that we have to find targets for them. So there is need for new compounds. So the source of the compounds, we have two sources, major: natural products and synthetic products. Natural products, everyone knows what it is. Synthetic products, there is the concept which is born on 1990, which is the combinatorial chemistry. Combinatorial chemistry is based on solid phase synthesis, parallel synthesis, diversity-oriented synthesis, mixture-based libraries. So my talk will be based on these four concepts: solid phase organic synthesis, parallel synthesis, diversity-oriented synthesis, mixture-based libraries. And of course, there is the computational chemistry, which is the virtual screening. My talk, my work in the lab is based on how we transform peptides to small molecule compounds. Peptides, everyone knows, they are a very good source of finding hits. There is no assay that you don't test peptides and you will not find hits. But peptides, they have a problem. They have this enzymatic degradation. C-peptidase, N-peptidase, chymotrypsin, trypsin. It means it's very difficult to make a drug out of peptides. So peptides, what they have in particular, they have these side chains of amino acids. On the other side, when you check the majority of existing drugs, they obey to the Lipinski rules of 5. Lipinski rules of 5, which is, so molecular weight is less than 500. Number of hydrogen bonding, less than 10. Optimal is less than 5. Number of hydrogen bond donors, less than 5. And rotatable bonds, sp3 bonds, that are less than 5. And of course, the solubility, the pK, is less than 5. That's what we call Lipinski rules of 5. So if I go back here. So combinatorial chemistry, where is it? Okay. I will talk today how we transform peptides to small molecule compounds. So we gain two things. I maintain the side chains of the amino acids, which they are responsible of the activity. And I transform to heterocyclic compounds, which they are, for bioavailability, they have drug-like properties. When I say combinatorial chemistry, it's, let's say, this quinoline. Combinatorial chemistry is we have to try to do all possible combination of existing drug, let's say it's the quinoline, how we can make them. You will see that we use T-BAC technology, I will explain later on. We start from peptides and we transform them to a variety of heterocyclic compounds, cyclic urea, cyclic guanidine, diketopiperazine. All of them, they are drug-like compounds. And all of them, they originated from peptides. So I want you to focus on these numbers. That's the most important things I want you to learn. How we can make 450,000 compounds around N-acyltriamine. How I can make 472,000 compounds of this cyclic urea or cyclic thiourea. There are compounds that we are making like this acyl bicyclic guanidine. We make 1,300,000 compounds. So this is the concept of libraries. How we make them. That's why the thing I want you to learn. So this is chemistry examples. I will go very fast. How we transform peptides after chemical transformation to heterocyclic compounds. This is another example. So I give you this example for the parallel synthesis. If I start from resin-bound acylated amino acids, after reduction, I will generate this resin-bound diamine. So when I treat it with B-functional reagents, like here oxalyl dimidazole, I will make the cyclic urea or cyclic thiourea. With cyanogen bromide, I will take this cyclic guanidine. Here the diketopiperazine, here the piperazine, here the triazine dione, and here the diazine dione. So if I start from 15 amino acids and 15 carboxylic acid, I will make 225 compounds of each heterocyclic compounds. With the technique that we have in the lab, parallel synthesis, we can make these, all these compounds, 225 compounds, of each heterocyclic compounds, we can make 225 in 2 months, maximum 3 months. So every heterocyclic compound here, we are making 225 compounds. I'll get to that. Yes. So if I make 3 diversities, we will increase this number actually to hundreds of thousands of compounds. So this is isolated from dipeptide. We block this amine. When we do the reduction, we have still diamine, and then this amine will not react, and we'll make all this variety of heterocyclic compounds. This is how we make bicyclic guanidine. So what we have at the end, as drug discovery lab, we have diversity of scaffolds, and around every scaffold, we have diversity of functional groups around each scaffold. So we have a good background of structure-activity relationship. So we will have the optimal scaffold having optimal functional groups. This is an example how we transform tripeptide to bicyclic compounds. So like this one, we made hundreds of thousands of compounds, and following the screening, we found compounds like this for the opioid receptor. So they are more selective for the kappa and delta. They are not very, no, actually they are very selective for mu, and then kappa and delta opioid receptors. So I have to skip many things. So this example is a very good example, the synthetic of hexahydrodimidazodiazepine from resin-bound tripeptide. Following reduction with the borane, we have this 1, 2, 3 triamine, that when we treat the triamine with this malinyl diimidate, we will find these fused diimidazodiazepine compounds. This library means we published lately on Journal of Medicinal Chemistry. They are very selective for kappa and delta. And what is good about this compound, when they react with this opioid receptor, they react on the peripheral. They don't go in the CNS. And then when they act only on the peripheral, they don't cross the BBB. What will happen? All the side effects, not all, most of the side effects associated with opioid receptor, we will eliminate most of them. Constipation, the most major problem is the addiction, and respiratory problem. We will be eliminated. Why? Because that compound did not cross the BBB. It's only acting on the peripheral side of the brain. So here there is an example how to find from mixture-based library, large libraries, hundreds of thousands of compounds, compound to treat cancer. To find a compound, active compound, we need this phenotypic versus target base. So we screen compounds, we don't know any idea about them. This is the phenotypic versus target base. So phenotypic screening. We make a library, we have no idea if it will be active or not. So the assay it will tell us if we find active compound or not. And then the other one is target-based libraries. It means you know through X-ray or modeling that this compound, it will react with the receptor, and you do the synthesis. So this is target libraries. So like here, an example to treat AKK epsilon, it's a project with someone, University of California, San Diego, to treat cancer. So he came to us, to my lab, and he said, we need compounds of this class. Why? Because when we did the computational studies, they seem to interact with the receptor better than the existing drugs. So we have to find a strategy. So how to make these compounds? So I will not go through the chemistry because it's very, okay, it's not the goal. I want to show you how we make compounds. So these compounds that we made a library, we found very active compound to treat cancer. And the same library, actually, it was screened. So a very active compound to treat AKK epsilon. And this same library, it was screened against Alzheimer's disease. And we found very active compound to increase the protein SSAP alpha. By increasing this protein, you will give and revive the cells in the brains. So these two compounds, they are actually covered under patent right now. So I will skip all what is cyclic peptides, and I will focus because I want you to know how we make these numbers I mentioned. How we can make 450,000 compounds. How we can make 1,300,000 compounds. We screen that library, and still we can find active compounds out of that. So the idea is mixture-based libraries. So when you check the nature, how we find drugs? We find drugs from extracts. When you have like the green tea, when you can hear my voice, it's loud, yeah. Yes? When you have the green tea, and you drink the green tea, and they tell you there is medication in it that it will relax you. So the green tea is an extract of natural product. In that extract, there are hundreds of thousands of compounds, yes? But it's only one substance which is responsible for the activity. Yeah. So the message that screening, what we learn from nature, is when you do a library, you don't need a pure compound to test it to find the activity. You can screen the compounds, the chemical libraries, as mixtures. And then you have to find a way how to find the active compounds. That's the mixture-based libraries. So, natural products. 30% of the drugs that come out of natural products. So I have to skip this. So again, when you have an extract which is active, and then you know that extract is active, you have to do all possible extractions. You do many fractions, and then you go to those fractions, you separate them, and then at the end you have an active compound. Yes? So you know that the activity comes from them. So you will give to the chemist NMR, everything. They have to determine the structure. Most of the time they will not be able. The message here again, you don't need one pure compound, non-compound to have a drug. So, how we can make 6,250,000 tetrapeptide? All of you know what's tetrapeptide. We have 20 natural amino acids. There are hundreds of non-natural amino acids. So if I take the known 20 and their equivalent D amino acids, and I have extra 10 amino acids, non-natural amino acids, I have 50 amino acids. How I can make 6,250,000 compounds and still I have the capability to screen these 6 million and I be able to identify for any assay which is the tetrapeptide among 6 million peptides, which is the tetrapeptide that is responsible for the activity. Yes? So, I have 6,250,000 compounds. So, how we do it? We make the library, the tetrapeptide. We make the synthesis of the library 50 times. So how we make these times? We have to do all combination. 50 times 50 times 50 times 50. It's 6,250,000 compounds. If I have to fix the first amino acid, I want you to understand this. If you understand this, you understood the concept of combinatorial chemistry. I fix the first amino acid, and the second amino acid, third amino acid, and fourth amino acid, they come all mixtures. All mixtures. So I will mix 125,000 peptide here. So for every single mixture, I'm having 125,000 compounds. So here I will have 50 vials. In every vial, I know what? I know only the first amino acids. I do the synthesis again. Why I only know the second amino acids. I do the synthesis again when I will only know the third amino acids. And I do the synthesis again when I only know 40 amino acids. I will ask you a question. If you give me the answer, that means you understood what I am saying. So how many vials do I need to make? It's 200. The answer is 200. Because I need to make 50 here, where you define only the first amino acid. 50 here, the second amino acid is defined. 50 here, the third amino acid is defined, and 50. So I'm making only 200 vials, and by doing the screening assay, it will give me an answer for how many? 6,250,000 compounds. You are asking how? If I take the first set of 50 amino acids, okay, I will screen them here. It's kappa receptor binding assay, just an example. So I'm having the first position is defined, which is O. X via all mixtures. So here I know I have 50 vials. What I know in these vials, I know only the first amino acids, yes? I screen them. So it's telling me that the vial, that the first amino acid which is known, like alanine as an example, valine or serine, I see activity in it. The second 50, I know only the second amino acid. Okay, thank you. Okay, I know only the second amino acid here. And here I know only the third amino acid. And here I know only the fourth amino acid. So what I have to do, this one is no interest. I did not see any activity. So I take 1, 2, 3, 4, 5, 6, 7. So for the first amino acid, there are 7 mixtures. They showed activity. The second position, there are 4 amino acids. They showed activity. The third, it's 4 amino acids. They showed activity. Here, 2. And here, the last position, it's only 2. So I will take this one and this one. They showed some activity, yes? So what I know, here I know the first amino acid. Here's second amino acid, third amino acid, and fourth amino acid. What I have to do, it's only the combination of these active mixtures. So the activity, I screened how many compounds again? 6,250,000 compounds. The activity right now, it has to be within 224 compounds. It has to be in these 224 compounds. I have to make these 224 compounds and then test them. So we tested all of them, and this is the most active compounds. This is a very beautiful story. You have to understand this. So what we found at the end, a combination of D amino acids, 1, 2, 3, they have very strong activity, 0.7 nanomolars, kappa selective. This compound is in clinical trial 3. So it will be a drug. It means next year or the following year. What's important this, I cannot think of any other possibility to find this peptide, all D amino acids. More than that, they are non-natural amino acids. There is no other way that you can find a drug like this. This tetrapeptide is on the market, and it will be on the market by next year. It's the clinical trial 3. And it's acting, again, at the peripheral side, because peptide, they don't cross the BBB. So the advantage as well, it will have a very limited side effects known for the opioid receptors. So this is how you make, again, the message for combinatorial chemistry. You don't need to have a pure excess enantiomeric 99% compound to be tested to find a drug, yes or no. No. You can work with the mixtures as long as you have a strategy, if you see any activity, how to chase that active compounds. And that's the positions. This approach is called positional scan approach. So I'm very sorry for the time, yes, but any question? Yes, just 1 minute, please. Why? Actually, I will show this. I cannot leave without showing the Tunisian flag. So I have to thank all the coworkers that worked with me. I had the chance to train all these PhDs as a postdoc. So they have done the job. So my lab is very open to actually Arab scientists, Arab students. What I learned from today's message, do not blame the politician, no. If you can do something, you can do. If you have the desire to achieve something, you can. Yes, our system in the Arab world is corrupted, and you know it. But if you know it, you cannot stop there. I was very impressed with Miss Hayat, her name. Yeah, look what she did. Saudi, raised in Saudi country, as a girl, very conservative society, and still she went and they refused her and she persisted. Look at her, what she's doing. So everything is in your hands. Don't blame the politician because nothing will change. But you can do things. أقول قولي هذا واستغفر الله لي ولكم فاستغفروه.